Good afternoon, my name is Kyla Sinat, and today with my teammate Alisa, we're gonna explain how we identify an unknown bacteria using both traditional microbiology technique and also AI assisted analysis. The goal of this project was to determine the identity of an unknown sample through a series of biochemical tests and observation. In the rest of the video, we're gonna talk about our process, the different results, and our conclusion. The goal of this experiment was to determine the identity of an unknown bacterium by collecting data on its morphological and physiological characteristics. We begin by observing colony morphology and performing a gram stain. Our results showed that the bacterium was gram negative, which immediately helped narrow down the list of possible organisms. Based on the given possibilities, we considered both coliform bacteria such as E. coli, Klebsiella pneumoniae, Enterobacter aerogenes, and non-coliform bacteria such as Salmonella typhimurium, Shigella flexneri, and Pseudomonas aeruginosa. Our methodology was the investigation utilized biochemical testing to identify an unknown bacterial sample. The investigation began with gram staining to determine cell wall characteristics, followed by lactose fermentation testing to evaluate metabolic activity. The bacterium was identified as gram negative and lactose non-fermenting. Further testing included methyl red and urease to assess additional biochemical properties. Along with a motility test, we determined movement. The results from each test were used to guide progression through a dichotomy scheme for accurate classification. Based on these findings, the unknown bacteria was identified as Shigella flexneri. Overall, bacteria identification was achieved through a structured process combining biochemical testing and systematic analysis. Our result, what did we get from all of this? Gram staining revealed pink, rod-shaped cells indicating the bacterium was gram negative. In the lactose fermentation test, no color change was observed, indicating a negative result for lactose fermentation. The methyl red test produced a red color, indicating a positive result, while the urease test showed no color change, indicating a negative result. The motility test showed growth only along the stab line, and this indicates that the organism was non-motile. These results are consistent with Shigella flexneri. Overall, differentiation staining and biochemical testing allowed for accurate identification for unknown bacterium. Next, what we did, we then use the dichotomy scheme, which is the tools that allows identification by answering a series of two choice questions. Starting with gram negative result, we follow each step based on our chemical test result, and until we were left with the final match, Shigella flexneri. In addition to all the traditional lab methods that we did, we also use artificial intelligence tools to support our analysis. One tool we used was ChatGPT, which helped us interpret biochemical tests and compare characteristics of different bacteria. Another tool we used was an AI-based image analysis tool, which helped analyze observation and confirm morphological features. And we also use specific prompt. We ask specific prompt to AI. For example, given a gram-negative non-lactose fermenting, methyl positive, urease negative, and non-motile bacterium, what is the most likely organism? And this type of specific prompt gave us accurate and useful result from the AI. Also, to verify the accuracy of the AI generated responses, we compared the result with our lab data, our textbook, and also our dichotomy key, just to ensure that the suggestion made by the AI match our experimental findings and were scientifically correct. What this project helped us it helped us become more knowledgeable about both microbiology and AI. We learned how different biochemical tests contribute to identifying bacteria and how AI can assist in analyzing and interpreting data more efficiently. These are very useful in future studies and careers, and where technology is increasing and we can use it alongside in lab work. In conclusion, traditional microbiology methods prove effective in identifying the unknown bacterial sample. So the bacteria was determined to be gram-negative, non-lactose fermenting, methyl positive, urease negative, and non-motile, leading to its identification, which is the Shigella flexneri. The step-by-step use of biochemical testing and with the help of the dichotomy key allowed for accurate classification of the organism. These findings highlight the reliability of classical microbiologic techniques in bacterial identification and also their importance in both clinical and laboratory settings. The results were also supported through systematic analysis, reinforcing the accuracy of the identification process. Further research and acknowledgement. Future research could explore additional methods to improve bacterial identification, especially in cases where biochemical results are unclear or they're overlapping. Incorporating molecular techniques such as DNA sequencing could provide more of a precise identification and reduce uncertainty. Expanding the range of biochemical tests may also help differentiate closely related species. Additionally, integrating computational tools or AI-based analysis could enhance accuracy and efficiency in identifying unknown bacteria. Combining traditional laboratory methods with advanced technologies may improve reliability and support faster decision making in microbiological studies. Thank you for watching our video today. Let us know if you have any questions.